Okay, so before we start, I'll just repeat the important information about the course. There's students that sent me an email saying they're not going to be here, but apparently only one of them showed up, so there's that already. That's good. I'll also show you actually the course outline first, and then we'll get into the course. This lecture is quite short. Well, not short, but you know, I said that last time, and then we ended up doing it full time. But it will be quite simple, and then starting next week, we'll get into the nitty-gritty of genomics. Okay. For those that are taking 512, you will see that the courses are kind of complementary, but sometimes we'll be talking about the same stuff, so that's double the practice for you. But I try to avoid copying Amandeep because we're teaching genomics, and sometimes there's a gray zone between functional and structural. Okay, so first let's try this. It's not working. We got this. Oh god. Okay. So I changed my mind for the meetings, for the office hours. So I was going to do it by Zoom, but I realized, or by appointment, but I realized that every student will ask me at a specific time, and then I end up doing 20 hours of, you know, office hours. I would love to do that. I actually don't mind staying longer. I'm not like those profs where, like, you know, I gotta leave. So I'm okay with that, but let's just schedule. What the hell is happening here? Okay. Let's just schedule it right after today's lecture for 1 hour, and then if you can't meet on those times, you can send me an email. We can do a Zoom call. I don't care if it's the evening. I don't care if it's the weekend. I'm always working, so just send me. We'll figure it out. Okay. The reason why I try to do Zoom meetings, it's because I'm between two campuses. So for me to do 2 hours for a 10-minute meeting is not worth it. But if I have to do it, I'll do it. No problem. Okay. So there's that. For the course content. So this course is organized in a logical way, easy to follow. So we'll start by talking about the structure of a genome in a base model organism, which is the yeast, the simplest eukaryotic organism that we usually study in the lab. That's why we chose yeast. And then we'll take a look at how we get the information from the yeast genome, and then we'll jump to the human genome directly, see the structure of the human genome, which is pretty much the same thing, just more proteins involved and more longer segments of DNA, let's say. And then we'll take a look at some interesting stuff like the Human Microbiome Project, which will tell us some stuff about the human genome, which will tell us about the properties of the human genome. So this part is quite interesting. The first part is kind of boring, and then you get into the actual fun topics. Then we'll take a look at genome diversity. We'll look at the Neanderthals genome, which was sequenced by Svan Pablo. And then we'll take a look at some genome applications. Maybe I might change gene therapy to something else, because I see here that I'm not talking about ecology or bioengineering or all that. So maybe I'll switch up this one here. But cancer, for sure, we'll talk about it because it's highly relevant to us. This one too, but you know, gotta diversify. And then at the end of the semester, there's 3 lectures where students are going to present. They're going to present on topics related to genomics. I was told by Dr. Whiteway, so Malcolm, to kind of choose the articles for you, because students tend to not choose the right level or just, you know, mix it up and get lost. So I'll see what I'll do up when we get there. Maybe I'll ask you to choose a topic, and then I'll get you an interesting article about it, and then you can present about it. Okay? We'll see. I have to see what the hell is happening until then. So for the coursework, who's taking 512 already? Okay, you guys are going to die this semester. I'll tell you that right now, because I just looked at Amandeep's outline. And so you basically have pretty much 2 presentations, 4 papers, 4 homeworks, 4 quizzes, like all together, okay? So in this class, at least I looked at the grades of Whiteway, because I'm copying his class almost completely, at least the first few lectures, okay? And then I'll add up from my stuff. But the grades were pretty good. They were between B and A. There was no C, no D in the class. Again, this depends on the level of students, and also the time that he taught it was in COVID, so you had access to pretty much anything. So I don't know if that's going to decrease this semester, but there's ChatGPT too, so you know, I'm pretty sure you can figure it out. But what did I want to say? So there's class participation. The reason why I'm putting a grade for this, it's because I want you guys to get involved. This is a small group, so you need to get involved. You'll have to figure out who are the good students to get in teams with to not fail your presentations, so might as well do it now, you know. You also have 4 homeworks. The first one was given last lecture. I'll show it to you now. It's just a homework that you have to write something quickly. You can use ChatGPT if you want, but if you want to actually learn and figure things out by yourself, you could do that and then fix your work using ChatGPT. Okay, I have no problem with that, because we have to start using it. Otherwise, you're going to lose the edge, okay? And I don't care what other professors say. It's part of the game, okay? The only thing that I have to say about ChatGPT is that I've been using it since it came out. It's very good for coding. It's very good for calculations. It's very bad at, you know, telling you information about facts, people, and time, okay? And so since we're working with papers, well, you might have problems getting the facts true, okay? I know that some of you will copy the whole paper, put it on ChatGPT, and spend so much time editing the PDF into a Word document and trying to figure out how to write a paper about that. You'll see that ChatGPT is limited in terms of how much it can write, and then you waste more time on doing that, because I've been using it for 2 years, okay? I even have a friend who used it to get his MBA, okay? His thesis for MBA, he wrote it just using ChatGPT, okay? It seems illegal, and universities are against it, but it's part of the game. You have to use it. Like, this is old school mentality that, like, you should not use the tools that are available to you, okay? So I'm for that, but be careful. Be careful, that's all I'm saying. And then ChatGPT sometimes gives you hallucinations. So these are just things that seem true, but if you have a little bit more knowledge, you find that it's false, okay? So there's that. The other thing that I want to say about it, actually I have a lot to say about ChatGPT, is that usually when you invent something or you discover something, you embellish it, okay? I'm going to cure aging, I'm going to cure cancer, we treated this. And so with ChatGPT, they use this artificial intelligence can do everything for you, right? Reality is they're doing t-tests, they're doing clustering analysis, so that's just statistical tests, okay? So when it's generating that information, it's just doing tests to see what's the most probable thing that could appear after. Not the most probable in terms of true information, the most probable that it's seen in the information that was used to train it, which is half of the biggest library in the US. So that's not a lot, okay? Especially in terms of science, that's super complex and very hard to predict, okay? So that's my ChatGPT part. The midterm exam is quite easy from what I saw in his midterm exam and final. Obviously, I'll change it a little bit, just, you know, depending on how the course goes. There's no multiple choice, no true or false, none of that, okay? It's all writing. There's maybe 8 definitions, I think, or something like this, and then the rest is like text that I need to see you thinking, okay? Obviously, I'll do it for fun, but you can figure out who actually wrote their assignment and who didn't just by comparing the midterm exam to the assignment to see if it's the same writing pattern. So there's things like this. I don't care. It's not going to change your grade. You can use ChatGPT if you want, but I'll do it just for fun to see what's happening, okay? Final exam, it's a bit longer compared to the midterm, maybe 1 or 2 questions, additional questions, but that's it. And obviously on the topics that we cover in the lecture, articles, etc. Okay? Oral presentations, you'll be in teams of 3, I believe. 3 or 4. I'll see for the oral presentations, but basically you'll have to present a paper that's either assigned to you or that you choose, sometimes 2 papers to complement each other. And it's just to see how you're able to understand the paper actually, and then just communicate what's in there, and then, you know, discuss limitations and things like this, okay? This is typical from 400 level classes, 500, 600. Actually, 600, you'd have a bit more work. Anybody in PhD here? Okay, good. Less work for me. Usually, the professors, they like to give different grading schemes for undergrad, masters, and all that. I think you guys are not far from each other, so it will be the same. I will not give you longer reading assignments or longer midterm exams or whatever. Not because some people might think that this is lazy or whatever, but actually, no, don't think that your level is much different from a 400 to a graduate diploma, okay? Further than that, the professor that gave this course here, Malcolm Whiteway, has been in the department for a long time. He's a tenured professor, and he does it this way. So I'm just going to follow him, because I'm still learning also, okay? Okay, so the homework. Very simple homework. Just take a paper from the press, not from Nature, Science, and all those scientific articles, and just tell me the story about it and why you chose it. Try to get like a paragraph, maybe this big, I don't know, 100 or 200 words, something like this. And then this will basically just tell me how you write, so that we can correct that for the bigger assignments. That will be like 4 or 5 pages. And you'll see how I correct work, okay? So this is a test, and then from there, they will start counting. I forgot to mention something here. So class participation, this is, we'll have discussions during lectures, usually around topics that are a little bit more political, but related to the science. So to answer it, you need to use science, linked to whatever topic that we are learning that day, okay? Cool, so let me drink some coffee so I can see clearly, and then we can start, okay? So, let's start by the basic yeast genome that you heard about like 2,000 times, right? So why do we use the yeast genome to study structural genomics? Well, for the same reasons you've seen in the past. Yeast is a very small organism. It looks like this. This one here is dividing into 2, budding. It has a big community that was invested in it. Now it's a little bit dying off, kind of, because this is like 1970s, 60s, 80s. I did my PhD on this, so I know it pretty well, but the genome is small, so there's 12 million base pairs, which makes it very easy to work with, and most of it is genes. So the ratio nucleotide to information is actually pretty high, so you're not dealing with a lot of junk DNA, which is quite hard to study, especially when you don't know what's in there, right? Because you have to think of it as, at that time, we know that DNA exists, but you don't know what the hell is the makeup of DNA, right? And so you're trying to figure it out. You don't have the structure of it, and so to find the structure, you're kind of blind, right? So you need something that always gives you a trait, and so you can kind of study that trait functionally, so that then you know that there's something there that does that, and do it by association, okay? And we'll see that in this lecture. There is also this important feature here where previously scientists mapped the genome by classical genetics, and so they knew where everything was. Classical genetics means you mutate, and then you see what the hell happens to the cell, and then you repeat for all the 6,000 genes, and then you know what they all do approximately, and then they have, binary functions, etc. But that's further on in the class. And the most important of all this is the fact that you could play with the genome easily. So yeast can be haploid or diploid, meaning if you knock out the gene in a haploid, the effect is immediate. If you look at humans or other organisms, sometimes in the heterozygous state, what happens is that you will not see the effect because there's one that's complementing the other, that's contributing for the effect, and so it's sufficient to prevent the effect of that mutation. Am I going too fast? Okay. If I go too fast, tell me, because sometimes I tend to rap, and I forget that I'm teaching, okay? So transformation allowed for, a single transformation allowed for genome modification. Let me put my laser. And we were also able to make artificial chromosomes.
So if you heard about synthetic biology, it seems like it's a new field, but it's not a new field. It's been there for a while, since the 1980s. It's just now it exploded because we realized that we can do a lot of things with it, like taking sugar and transforming it into biofuels, things like this. But we were able previously to create these artificial chromosomes that contained genes and then put those chromosomes inside the cell. They did not work perfectly, but they worked, okay? So now you know why we're studying yeast. We'll later on extrapolate to the human genome, and you'll see that it's pretty much the same thing, just a higher degree of complexity because things multiply in the cell. So the proteins involved multiply, the structures multiply, and things like this, but the base remains the same. Actually, sometimes when you're studying genes, it's better to study it in yeast. Like, for example, aging. It's very hard to test on humans drugs that extend lifespan because you'd have to give them the drug, wait for them to die, and then see, right? So that does not make sense, right? So what you do is you test it in smaller organisms, and usually the genes that are involved for aging are the same everywhere because they are essential genes to survival and proliferation. And so the TOR gene, for example, is the same everywhere, okay? And so this is actually part of what I was doing during my PhD. I was studying aging. And so instead of studying it in mammalians, I was doing it in yeast. And then when you figure that out, then you move to mammalian cells and then mice, etc. Okay? I'll tell you a bit more later about my research, but first I need to give you some information. So if we take the yeast chromosome, there's only a few parts that you need to remember for in terms of structure. You have the genes, obviously. Now, why aren't we doing this in bacteria? Because we're interested in eukaryotic genome, right? We're interested in learning all this for human benefit, not just like that for fun. Okay. So there's genes. There are these regulatory sequences like enhancers, if you remember from your genetics class, or probably other classes, the gal operon. So gal 80 is an enhancer. No, it's a transcription factor that binds the enhancer, which is the upstream activating sequence. So there's these regulatory sequences that are upstream or downstream of the gene. Proteins bind them, and then they change the conformation of the DNA in a way where the polymerase can do its job. Okay. Sometimes they block the polymerase, etc. There is selfish DNA. Anybody know what selfish DNA is? No? Oh, yeah.
There was a book named after the selfish gene. Is it?
The selfish gene, though? Yeah, I think I have it at home on top of that.
I'm not completely sure. I believe it's tied to the way genes are reproduced. They will try to maintain their own material instead of trying to combine with others.
Exact. I like this guy. So selfish DNA. Okay, so that's the evolutionary perspective, okay? So there are some genes that will tend to stay in the cell for a while because they confer some benefit. But selfish DNA is just, you probably heard another name called satellite DNA or transposons. Transposons, anybody? So these they basically just improve their chance of being transmitted, but there's no evolutionary benefit, no natural selection benefit. So they're just selfish, okay? So we'll see these. There are centromeres. This you probably know. In your chromosome, you have the centerpiece there, which is important for spindle fiber attachment and then pulling the chromosomes to separating cells. Telomeres, which is the end of the chromosomes. These are important for many things, including aging, cancer, and other stuff. And so this is a distinct section of the chromosome. There are replication origins. These are important for replication of the DNA. So you can't replicate everywhere during the chromosome. There are specific places where you can do that. So this is a structural feature of the eukaryotic genome. And then there are finally functional RNAs. Functional RNAs are ribosomal RNA, tRNA. I think someone was working in Joy's lab
told me last week, exactly. So you were working on tRNAs. So this is a feature of the genome. So we'll see about these guys. Let's take just a second break because I remember that you asked me a question last time. So I did Google the stuff and I looked it up. The question was if only 2% of the genome was actually coding for traits, then the rest is junk. And so how is the forensic team using SNPs, so single nucleotide polymorphisms, to determine differences between people and if it's a criminal or not? Okay, so Akil, right? Akil was right in a sense. He said 7 SNPs are used, but actually it's not 7, depending on the, I think that's old information maybe. I looked it up. There are some teams that use about 90, so it's between 20 and 90, right? So you can see how the probability of that is almost impossible to find the same combination of SNPs if you're doing 20, right? To answer the second part of the question, which was about the 2% of the genome, so actually the SNPs don't look at your traits specifically. They look at the whole DNA, because even though the traits make the differences between you, even the junk DNA is specific to you. Does that make sense?
Yes, it does.
Good. So just a random question, not related to the exam, but had to figure it out. Okay. Let's see. So now we know that these are the pieces that are found in the genome. So now the question is, how the hell do we identify them? So we know that there's a chromosome in the cell, but we don't know that it's all A, T, C, G, A, right? So how do you figure out what is what? Anybody has a plan? No? Yeah?
Was something about attaching probes to certain parts of the DNA and then looking out for the fluorescence of those probes.
So when you're attaching probes, that means you know the sequence. And if you know the sequence, you know the structure.
That's true, because you need to know where to bind the probes.
Exactly. So now you're blind and you need to figure it out. How the hell do you do that? Okay? So new information for you guys. You use something called a functional assay. Functional assay is a very simple way to say, I will break something, I will see what it does, and then I'll associate that part that broke to that function. Okay? So this is what was used early on because we didn't know what the chromosome looked like, where the genes were, where those enhancers, silencers were. And so you start breaking it, and then if you see a change in the phenotype, you know that that part is important for that phenotype. Okay? After you've done that, then you can take that and find the structures using probes and things like this. Okay? So today we'll cover the functional assay part of it, because there's 2 more lectures on this. And then we'll take a look at the next obvious part, which is the structural approaches. Just one little paper here that I want to talk about. So this paper here, so we're talking about functional profiling of the yeast to identify the structural elements. So this is where your other class ties in. You'll see all the techniques that are used to do that. But I'll go over them quickly here today. This paper here was published in Nature in 2002, I think. And this is the paper that basically figured out what every gene does. Okay. Just to highlight a few names, you can read this paper to see how simple it is. But now it's very important in the field. Just a few names. This was a group of, I think there were more students, but probably they didn't include them. It's about 100 researchers that worked on this from 7 different universities: Belgium, Baltimore, Stanford, so Cali. Concordia participated, and who else? And McGill. Okay, so Montreal is on the map. We're handling some stuff there. There's a few people that are important. Reginald Storms. So this used to be my professor in 2012 or 13. He's retired. This guy is a G, very nice guy. There's also Marc Lucier. Where is this guy? He's right here. So Marc Lucier used to be a student at the time. Now he's the director of the Neuro at Montreal. So there's some big names here. You can take a look at them, and you'll see that I talk a lot about people. Not because I like to remember people's name, but because you'll see that this field is a lot about knowing people and the names. It's not really about the degree and all that. So it's important to know who's doing what, so that you can move forward, let's say, okay? But yeah, his office used to be right in front of my master's office, so I would see him every day. He's a pretty nice guy. Just relax, always walking slowly and thinking about life, okay? I don't remember, I don't think nobody got the Nobel Prize for this, I think. Okay, so let's start by the first part. How do we identify genes? This is the one that you know the most. So genes, usually what they do is that they produce a protein, right? And usually it's an enzyme, sometimes it's something else. So what you do for genes, you need to do a functional assay related to the function of that gene. If, for example, you're looking at an enzyme that converts pigment A to pigment B, then you're looking for a change in color, right? So you're looking for that function. So then when you break parts of the DNA and test which one confers that, then you'll see this one actually turned it into purple, so this must be the gene for turning blue into purple, okay? Very simple way to explain it. We'll get into the details in the next lectures. The way you do this is through cloning. So there's cloning in bacteria, so we're going backwards. So yeast is a eukaryote, bacteria is prokaryote, but the genes, sometimes they work in bacteria because they have the same enzymes, okay? Glucose, let's say hexokinase, which is part of glycolysis, is not changing from here to here, okay? It's the same one. There's just more copies of it. Enolase, for example, exists here and exists here into, I think, 5 copies here and just 1 here, okay? So there's that. And then when you have these two, then you do the probes, which our friend was talking about. So then you can, when you know the sequence from there, you can use the probes and figure out, you know, what's happening in your cell using those probes. Cloning. This term mixed me up a lot in my studies. So I'm going to clarify it here. When we're talking about cloning, we're not talking about cloning people or things like this, like the sheep dolly. Mr. Ian, something, I forgot his name, passed away, but this guy was the guy that took care of cloning the sheep dolly. You guys know this story, right? Yeah. Okay, it keeps coming back forever. It's the only one we got, you know. So cloning for genes, it's basically taking a gene and putting it into a vector, okay? So the vector usually is a plasmid, and that plasmid, you'll cut it, you'll cut the gene also, and then you'll put that gene inside the vector, the plasmid, and then you'll do some stuff with the new plasmid that you generated. Anybody doing cloning here? Yeah, you do? It's very annoying. Looks very simple, cut and paste, but no. The gene comes out the other side, sometimes it doesn't go in, and then you just spend like months doing that. Some companies are selling clones for like $2,000-3,000, so if you can start a biotech on that and do it better, that's a good idea, because some of the longer genes are very hard to insert into a plasmid, and so that's why they're very expensive. Now what they're doing is that they're just printing the whole plasmid directly. So you can 3D print the plasmid. Crazy stuff, right? But at the time, if you were able to clone one gene, that's it, okay? The lab where I'm working in, he cloned about 8 of the receptors that you'll see in your classes, the estrogen-related receptor, retinoic acid receptor. So these are like things that you see in your textbook pictures, okay? And by just doing that, you end up becoming a fellow of the Royal Society and killing it in life, okay? Now, that's not enough, okay? Now you need to do a lot more than that, but this is the basis of research in the 1980s, okay? Anything not clear here? Okay, a little bit about my research. I use cloning a lot because I work on single mutations in a protein called the estrogen-related receptor, which basically is a transcription factor. So I take a plasmid, cut, paste, and see what different mutations do. If it affects the ability of the transcription factor to bind to DNA, and if it doesn't bind, then there is no transcription, and then no proteins, and the pathway is all destroyed, okay? Why is this relevant? ERR, so estrogen-related receptor. Alpha, for example, is very important for metabolism. So if you play with its level, you can treat different diseases like cancer, NASH, which are already published, things like this. So I'm studying it in the context of triple negative breast cancer. And you'll probably see it if you ever do research. Everyone does cloning, okay? Well, unless you have a lot of money in your lab. We do, actually, but we're still old school. If you have a lot of money, just print the plasmid and that's it. Don't, you know, don't break your head. So, PBR, this famous plasmid that used to be the same 12 years ago, and it's still the same coming in the lectures. You guys probably know it. So this is one of the earliest vectors that was used to do this kind of work, where you're studying yeast genes into bacteria. So some important features, you have the origin of replication for bacteria, which RNA polymerase comes here and starts, and helicase opens it and then starts copying and all that. You also have the selectable markers, so usually it's ampicillin and tetracycline, and you have these cut sites where you can cut with restriction enzymes and then cut your also DNA fragments and try to put them in here. And then, let me just go here, okay. And then you put those plasmids into bacterial cells. So for the genes, you don't know that they're there. So the way you do this is you break the DNA into pieces. You can either do that physically, or you can do that using enzymes, right? And then you cut the whole genome into pieces, so you can see how this is a lot of work. And then all you do is put these fragments inside these vectors, separate the vectors from each other. So imagine you have 6,000 genes, and then you have to do, well, actually, no, not in 6,000 genes. Think of it as 12 million base pairs. So then you cut it into smaller fragments, and then multiple different combinations. So maybe you could start from 0 to 100, and then from 50 to 100, etc. And so you do that for the whole genome. Can you imagine doing this with 3 billion base pairs? It's like impossible. Okay, so this is why you need yeast, which has a smaller genome and about 100 scientists who are working super hard. Then you get all these vectors with your fragment inserted in it. And then you will introduce that plasmid into E. coli that's missing some gene function. So E. coli, we had already characterized its genes and what they do. So usually people just store that as glycerol stocks, and you know that this E. coli here, so people collect different species, not species, but different strains, okay? And so you put it in a bacterium that is lacking a certain function, okay? For example, you could take a E. coli that cannot survive without leucine because its LuB gene is missing. And then what you do is you put all these plasmids in it that you just generated and see which one allows it to live in leucine. So this is called complementation without leucine. And you know that that specific DNA segment allows it to live without leucine, and so it's complementing the missing enzyme. Does that make sense? Yeah.
So when you're digesting the genome to start with, are you expecting that on average each gene will be left intact at least
No.
once, so that your plasmid, so
Yeah.
it's possible that you just wouldn't get a leucine complementation if that had been digested originally?
Yeah, you could. You could actually not get it, but usually these experiments are repeated many, many times. And using this method, they were able to do that. There's also some other advanced stuff, which actually I'm not going to get into that. I'll show you later because I don't want to mix you up. But basically, leave it. We'll get there. We'll get there. So when you find these strains that survive now because they have those, there was complementation, you know that that sequence is providing you with the enzymatic function of this missing LuB gene. Okay. The first few of them that were found were leucine, histidine. This one was the TRP. I forgot which one was this, but the four are just basically the use of amino acids and nucleotides as sources of energy, etc. And so if you look at the plasmids, the early plasmids that were used, so not the bacterial plasmids, but plasmids for yeast, you'll see that the selectable markers are not ampicillin or tetracycline or things like that. They are actually the gene for leucine, so leucine synthesis, histidine synthesis, etc. And so this is because these are the first ones that were found, and they were used directly to create those plasmids. Okay, so these are the selectable markers. Probably know about tryptophan. The others are pretty much the same. What else? Okay, so then you identify that piece that confers the growth. The next important thing is that you need the whole genome, right? So then you need to build a DNA library. You probably heard about DNA libraries. So it's just basically all the vectors that contain all the genes that are, working out for you, and the others throw them, right? So your library usually will represent the entire genome as fragments in the vectors, and fragmentation, as I said, could be through restriction, digestion, or through just physical methods. Okay. What else? Obviously, the fragments it's broken down into a random or pseudo-random way. What I mean by pseudo-random is that if you use the specific DNases, they will cut between different parts, but it's still random because you don't know really if there's a gene from there to the other one. Okay. Good. So, questions? No? Not bad, not bad. We're almost done. So now when you have these pieces, so this is what I was talking about. So the first ones that were identified for yeast was UR3, which replaced the function of this PIR-F mutant. In bacteria, the tryptophan 1, TRP1, is the one that replaced TRPC, His3, His-B, and then LU-B. Okay, LU2, LU-B. Some of them you cannot, because yeast is a more complex organism, so you actually don't know what that gene does, because you would put it into bacteria, but because bacteria does not react really to it, nothing happens, and so you don't know what the hell that sequence does. So for that, you need to actually put it back in yeast to study what that gene does in yeast. And so you know that this one acts as the enzyme UR3 whatever. And so if you put it back in yeast and then do that effect, like remove uracil, does that work? Not, etc. Okay? Sometimes you even end up with fragments that have two genes. It gets complicated, okay? It looks simple like this, very eloquent way of doing it, but in the lab it's a disaster, okay? It's a lot of work. What did I want to say here? And I think I just said this, which is basically producing the yeast. Where's cloning yeast through the development? Yeah, so this is just to make the vectors for yeast. So these are the selectable markers. Okay. So that's all boring. The interesting stuff is when you find a gene into yeast, for example, or into, let's say, human cells, you can ask yourself, does that gene exist into simpler organisms so that you can study it in that simpler organism much faster and then go back to the mammalian system? For example, in aging, sirtuins are proteins that are important for epigenetic. They're linked to aging, and they're mostly studied in yeast. Okay, it's just simpler, but they have the same function in mammalian cells. Actually, the first anti-aging gene was discovered in yeast, and funny enough, I did research on yeast, but I forgot which one it is. I think it's FAS or something like this. Anyways, so there's a lab in Cold Spring Harbor, the Wigler lab, that was studying the RAS gene. Anybody know what this does? Probably. Okay, so RAS is just an enzyme that converts GDP to GDP. And this enzyme was found to cause cancer in humans. And so if you mutate it, and then you activate it basically through the mutation, then you will lead to cancer because it promotes unlimited proliferation, uncontrolled proliferation. Cancer is a feature of more complex organisms. It's usually in multicellular organisms. And so a crazy question to ask would be, is there a RAS gene in yeast? and what does it do? Because evolution started from simpler organisms to more complex organisms, right? So you would think that it's not there, right? But actually, the Wigler lab did this experiment where they knew that RAS was there. They created these probes that you were talking about, and they did something called Southern blot, which is just a gel that you add to it these radioactive probes against RAS, and you put the genome of yeast. And they actually found 2 RAS genes, not a single one, 2 of them. So then the question is, what do they do, right? So in yeast, they're important for stress response. In flies, they're important for cell proliferation and cancer. And then nematodes, very weird, it's for vulvar development. And then mice and humans, it's that, right? So you can see that if you're trying to study the RAS gene, in terms of cancer, you're probably not going to use the nematode or the yeast, but probably the mice or Drosophila, okay? And so you would start with Drosophila and then move it to mice and then to humans. There's a lot of research on RAS. It's a very important cancer gene. So if you read on it, you'll see. And so this is basically going backwards using what we find in yeast to study genes in higher organisms, okay? Okay, is it clear how we identify genes? The dirty way of doing it, okay? That's like old school time doing it. Let's take a look now at the origins of replication. So the origin of replication, again, you have to do the same thing. You need to find a functional assay that tells you that this is happening. So origin of replication is just a spot along the DNA. It's a structure along the DNA, the chromosome, that allows for opening and then replication of the chromosome. And so if you remember, if you cut the DNA into smaller pieces, you have to find the piece that actually makes more DNA, right? Because it's allowing for more replication, okay? So you still have that plasmid, you put it inside the cells, and now this plasmid has 2 origins of replication, not only 1. And so there's more copies of that plasmid being made. So this leads to what we call, in terms of phenotype that we're looking at, it leads to a high efficiency of transformation. What does this mean? It means that if you're putting a plasmid usually in cells, you'll find, and then you select for it using a marker in an agar plate or LB plate or whatever, you'll find that 5 or 6 colonies from the millions that you had in the beginning, okay? Because it's a low efficiency transformation, right? In this case, because you have more origins of replication, you're making a lot more of that plasmid. And so when the cells are dividing, you're passing it on to more cells. And so you'll end up with a plate full of colonies. So this is what it means by high efficiency of transformation. In those plates, you know that that piece of DNA could give you the origin of replication, or it could be something else like conferring more resistance to whatever antibiotic you're putting, etc. Okay? Then you have to do more tests to figure out if it's either or. The only problem with this is that it creates unstable transformants. What do we mean by unstable transformants? So the plasmid has more origins of replication, so you're making more of that plasmid. It doesn't mean that it's segregating equally between the cells, right? Plasmids for yeast have a centromere region, so they get pulled out with the cells. So imagine you're making multiple copies of this plasmid. You don't know if 10 of them are going in one cell and then 1 is going in the other. So it's highly unstable. You'll see variation between your phenotype, and it's very hard to follow, but it works. A good example was when the scientists that were working on this did the transformation of the TRP1 or the leucine gene, leucine metabolizing gene, into bacteria. They realized that they reacted the same way, right? But then when you put them in yeast, for some reason, the TRP had a lot more transformants than the LU2, and that's just because they were not segregating properly due to what I just said, okay? And so you know that the TRP1 plasmid that they found, actually on top of that had the origin of replication stuck to it. Make sense? Good. So now that we have the origin of replication, we can start doing some cool stuff. So first we looked at the simple vectors, the PBR 33, 32. There's millions of different vectors. One type of them is called the shuttle vector. This one is still used, so the PBR is not used anymore, okay, unless you're working in bacteria, and even then there are better ones. But the shuttle vectors, they do exist, and we do use them in the lab. So shuttle vector just means that you shuttle it into a specific type of cell, bacteria, to amplify it, and then you use it on the actual organism that you're trying to study, okay? So the features of these shuttle vectors, so these shuttle vectors were only invented after the discovery of the origin of replication. Why? Because you need that origin of replication, one for E. coli that you're using to amplify, so that the origin of replication allows for the transcription of the genes that are important for E. coli amplification. And then you also need an origin of replication for yeast, so that you express the yeast genes that are found on the plasmid, okay? And I'll give you a good example in research so you understand. But the features are the following. So first you need, so these blue ones here are for bacteria. So you have the origin of replication here, and then I guess transcription goes this way. And then you'll transcribe the antibiotic resistance gene. So when you put this plasmid in bacteria, you select it with this antibiotic resistant gene. So if it's ampicillin, you add ampicillin to your agar plate. Only ampicillin, only cells that have taken up this plasmid will grow. So that's the first part. Then let's say you're studying some gene into yeast. So now you amplified this plasmid, you extract it from the bacteria, and now you're going to actually use it in your model organism for whatever question you're trying to answer, okay? In this case, we're doing it in yeast. So you also have an origin of replication for yeast. It's called the ARS, autonomously replicating sequence, which is different from bacteria. It's just called ORI-C. Not going to ask you crazy questions about like which one is which and why, right? You can find that on Google. So in this case, when this plasmid is inside yeast, when you transform it inside yeast, replication starts here, because the replication machinery of yeast recognizes this part here, and then will make the leucine biosynthesis or degradation enzymes in yeast, okay? And then you have also the centromere region. This is important for yeast plasmids because they allow the spindles to attach to it and then segregate it between different cells. Clear? Okay, now the example in research. Should I do it? You know what, let's do it on internet so you can see. So add gene. So me, when I'm trying to figure out to study a certain gene, for example, I will go here and I will look for plasmid. And then, where are they? So mammalian. I'll just give you an example here so you can see what I mean. 8 plasmids. So for example, let's do pcDNA here. Okay, this is just a quick example so you can see. So this is the gene I'm interested in, ER alpha. Okay, so for me, I'm putting this what do you call it? This plasmid inside mammalian cells. Yep. Yeah. Is that better? Okay, let's try to see this stuff here. Actually, you know what? I'll go here. Okay, so I'm using not this plasmid specifically, but I'm using a plasmid related to ER alpha, and I'm putting it into mammalian cells. So I need first the resistance genes that are important for mammalian selection. They're not the same as bacterial. So you have here neomycin and kanamycin, right? These are for mammalian selection. So when I put this plasmid into mammalian cells, I need to put kanamycin and neomycin to select for it. If I'm doing bacterial, very simple, ampicillin,
right? And then obviously you have the origin of replication for bacteria, and then for mammalian cells, I'm not going to get into it because it's like a few different spots. But one interesting thing is that there's also, so this SV40 here, so this plasmid, you can actually use it also in viral transduction, right? And so you can use a virus to inject this plasmid into mammalian cells. So some plasmids have this feature where they have genes that are recognized by the envelope and all that, and then they will go and inject it into the bacteria, into the mammalian cell. Okay, so these shuttle vectors are used all the time. Close it, okay. And it's not just in this class for some theory, okay? Well, let's see, I had an example and it didn't show. Anyways, I gave a different example, okay? Now, why am I talking about these shuttle vectors? So you can create some functional assays using these shuttle vectors. When you identify a piece that is doing some sort of feature, you can then put it back into yeast and then study it fully to understand what it does in yeast. Because not all genes have a function, not all genes, yeast genes, have a function in bacteria. Some of them don't, and so you need to kind of put them back into bacteria and then see what happens. For example, the cell cycle, cell division cycle mutants, division between prokaryotes and eukaryotes is not the same. And so you can understand that it's totally different between both. So if you put your CDC protein gene inside bacteria, there's no effect. So you have to study it in yeast. So for example, here I'm just giving the example where you're so you have this mutation in a CDC gene in yeast that you collected previously. So you know that this yeast, if you heat it up, it divides. If you don't heat it up, it does not divide. And so you have all these strains, okay? It's not like you don't have them, right? So scientists sometimes when they make colonies, if they find one that has a specific phenotype, they store it. So you have these. So if you're studying these genes, is my laptop dying? My laptop is dying. 1 second, guys. We don't want to lose that. So what was I saying? Yeah, so if you are looking at a gene that could affect cell division, then you'll need to find a mutant that does not divide, right? And so you have these mutants that have these temperature-sensitive mutations. Okay, which one do I remove now? 2 seconds, guys. We try. Okay, we're good. I was scared I would remove the projector. Okay, let me try that again. Okay, I can explain second time here. So if you are studying cell division, okay, you have these yeast strains that you have identified previously that have a temperature-sensitive mutation, so that when you heat those colonies, they grow. If you don't heat them, they don't grow. So you know that it's in a gene that affects cell division. Okay, that's the first thing. So if you're looking for the piece of DNA that can do that effect of division, then what you do is you take all your vectors from the DNA library that you found and then put them back into a shuttle vector, as we've seen, and then put that shuttle vector in yeast and then heat it up, cool it down so that it's at the restrictive temperature. So this example is opposite, but same theory. You put it at the restrictive temperature, and then you see which ones still grow. So which of those, that strain that did not grow at cold temperature now grows, and you know that that shuttle vector, the DNA sequence in it, is the one that confers growth, okay? And it's probably part of the cell cycle. Make sense? Good. What's left here? Okay, so now centromeres. How do you identify centromeres in these yeast cells? So we talked before about the origin of replication leading to unstable transformants, right? Because some of them had a lot of plasmid and others not. So if you're putting DNA on a chromosome,
and then you put it on in those unstable transformants, if you see equal segregation, meaning that the yeast becomes more stable, so every colony looks the same, then you know that you have a centromere, because now on top of the origin of replication that is there, you're also adding the centromere, and so now they have the capacity of being segregated equally, right? And that will distribute them evenly between the cells, and that will make all the colonies similar. In the other example where you just have the origin of replication that is inserted, segregation does not happen equally, and you'll see some colonies super big and others super small, and that's because some of them are resistant more or can metabolize X, Y, Z more, while the others are not. Okay? So if you add the centromere, now it equalizes the cells. Make sense? Pretty cool stuff. If it was me doing this, I would have no idea. I don't know how these guys thought about this stuff, but it's crazy. We'll be looking at the centromere structure in the future classes because we're talking about structure, and so this will be important. Okay. Same thing with gene, introns, exons, all that stuff. Okay. Last slide. Okay. So telomeres. Telomeres are the ends of the chromosome. Usually, when you have replication that happens, you're going to replicate in two senses. And so if you're replicating in the lagging strand, you'll leave fragments at the end, which get degraded. So those Okazaki fragments, if you remember, we have to rebuild them to prevent this. And so there's an enzyme that is a reverse transcriptase called telomerase that rebuilds that end so that they don't get degraded. Because if too much of the telomere gets degraded, you start degrading genes that are important for function, and then you end up either with aging or cancer. There's a group that identified telomeres in 2009. Blackburn, Grader, and this one I can't pronounce his name, Zostek, whatever. This professor here, actually his postdoc is one of the professors in the building, Malcolm Whiteway, which used to teach this class, so he was working with this Nobel Prize laureate. Okay, so we have some big guys in the department. Doesn't look like it, but we're here, okay? And so when you're looking for telomeres, again, you're cutting the DNA in small pieces. You're looking for the piece of DNA that is at the end here. And what's characteristic about this piece here is that it's at the end. And so if you put it into a plasmid, it will turn it from circular to linear, because when you put it in the cells, they will recognize it as being an end, and they will treat it as an end. So they will open up the plasmid, okay? And they will make it into a linear form. Now, this is a top view. The actual molecular biology behind all this is crazy. You can do like a class on just this part here, but we're not doing that in this class. We're not doing a molecular biology class. We're just going through each structure and then how we identify it, how it goes to the human genome, etc. Cool? Questions? No? Yeah?
So the shortening of the telomeres can lead to aging or cancer.
Yeah.
I understand how it can lead to aging as it eats into the chromosome. How does it lead to cancer?
Right. So, so the thing that happens is that aging is kind of a complex disease. It's not even a disease. It's more than a disease. Okay. So we're starting to consider it as a disease, but it's the conglomerate of all diseases that come with time. Okay. Usually you don't die of old age. You die of cardiac arrest, of cancer, of diabetes, of things like this, right? So, for example, in 1940, you did not live beyond 40, 50 years old. And it's not because food was not good or whatever, or you're not doing sports. It's just because before you had these infections that just killed everybody, right? And so if you're lucky, you made it to 40 years old. As we developed antibiotics, we increased the time between death at 40 and at 70 and 80, right? And so what happens is that now your cells are accumulating a lot of damage, right? And so you're aging with time. So for aging, there's 9 hallmarks, right? One of them is basically senescence, or the opposite of senescence, which is unlimited proliferation, right? And so before you get to that aging, you will probably affect a gene that will lead to cancer, right? You understand what I mean? And that will kind of kill you, right? But aging in itself is just the degradation of the body, and whichever part of your body is the weakest is the one that fails first, right? So if you have a weak heart, you'll probably end up, that damage will reach a point where it will affect your heart and you'll have a heart attack. If your brain cells are weaker, let's say, then you'll end up with Alzheimer's, Parkinson, dementia, all that, right? So it's just which one is the weakest, okay? If your epithelial cells are the weakest, well, you'll probably get cancer, epithelial cancer, okay? So this is what I mean by cancer. Actually, depending on what gene gets, affected the most and where it is, it can lead to this or that, okay? Yes, so that's pretty much it. If you don't have any questions, yeah? Complementation. This here? Right, so, okay. There are genes that you can study in yeast, in bacteria, but not all yeast genes can be studied in bacteria because that function does not exist in bacteria, right? I'm giving here an example with the cell division cycle, because division between eukaryotic cells and prokaryotic cells is totally different, right? So if you're trying to study cell division using a yeast gene, you need to put it back in yeast so that a fragment of DNA that you're not aware of, if it's a gene or not, you need to put it back in yeast. And so for that, you need a strain that does not divide under certain conditions and then grows on other conditions, right? So these strains, we have them from before because scientists like to just play with it, and then when they find a special colony that is of different color or whatever, they store it, okay? So now you find the one that, for example, has a temperature-sensitive mutation that grows at, so here this is flipped up, okay? But it's the same theory. So it grows at warm temperatures. Depending on the mutation, it could grow also at cold temperatures, but whatever. It grows at warm temperature, so that's the permissive temperature, and then you have a restrictive temperature where it doesn't grow. So now you take that cell that you have, and you put back that piece of DNA that you think is playing a role in cell division. You cool down, you cool down the plate, and you will see that some cells will grow. That means that that piece of DNA is replacing the missing function in that yeast cell, okay? And you know that that gene is important for cell division in yeast. You don't know which one of the CDC genes, but you know that it's important for cell cycle. Wait, sorry, repeat that. So you knock them all out?
Yeah, so if like the temperature needs to be high
Yeah.
for it to grow, and put it in cold,
Yeah.
nothing's gonna grow.
Right.
So when you put like one little piece of DNA,
Yeah.
you need 5 of them so that it grows.
You mean you need 5 genes?
Yeah.
Why would you need 5 genes? You're referring to all the cell cycle stuff?
Yeah.
Well, that's the thing. So I'm giving just a simple example of like one gene because I don't want to make it more complex. But actually, you get into these situations where you need all the pathway inserted. Okay, it's just I'm giving the simple example. But obviously, you do these duplicate and triplicate transformations with different fragments. Okay, it's a brute force approach that is very tedious, but works. So this is what I mean by complementation by cloning. So you clone the gene into a shell vector, put it back into yeast, and see if it complements or not. But obviously you don't know. It's not black and white, like there's the start codon, this is the end codon, right? It's just this piece does that. For you to know, because that piece could be the gene that does that, plus DNA that is unrelated, right? And so for you to know exactly where it starts and ends, we'll have to get to sequencing. Until then, we're just saying, okay, this piece does that. And it used to be very dirty work, as we call it in the lab, because you have that piece of DNA, You don't know if there's other stuff in there, but you're using that piece to do whatever you want in the lab, and then you publish papers on that, not knowing what's in there. It's just you know that it has the leucine biosynthesis enzyme. The rest, we don't care. Okay? So that's it. That's it. I just want to do something very quickly to get to know you guys. Okay? So I know some people don't like this, but you know what? They did it to me. I'm doing it to you. So we'll just go like this. Just tell me your name and which program are you in, so that I can then direct the course more specifically to the class. For example, if I see that there's a lot of people that are in IT, then I'll talk a bit about AI, deep learning, neural networks, computational genomics, and all that. Okay? Things like this. So we'll start by you. Yeah. Graduate diploma. Okay.
Nadia Cristiano, and I'm in the graduate diploma program.
I'm Gabriella Fassi. I'm in the graduate diploma program.
Yeah.
I'm in systems and information management.
Cool. I like this. Yeah.
I'm Hasti. I'm in Life and Health Sciences, which
Okay.
is kind of like our program, but in computer department.
Okay. So you're like in computer science, like full. You're less biology, more computer science.
No, it's still both, but a little
Okay.
bit more computer.
Okay. Do you take like 248, 249?
Yeah, I
Okay. I see. Yeah. Akil. Okay. Mark. See, I started remembering names.
Mark, and I'm in the graduate diploma program.
Yeah. International student too, I think, no?
Pardon?
International student?
Yeah.
I stalked your LinkedIn, don't worry. How do you feel about the tuitions? Hurts? Gotta do it, buddy. Gotta do it. Yeah.
I'm Maria. I'm in systems and information.
Okay. Shauna. Hi.
I'm also in health and life sciences.
Okay. Okay. Okay, there's a lot of graduate diploma. Yes. Graduate diploma. Yeah.
I'm Tommy. I'm comp sci in health and life sciences.
That's it. I like this guy. Go.
Alex, I'm also in systems.
Systems. Yeah.
Rachel. I'm in cell molecular biology.
That's it. Finally one from the bio. Yeah.
I'm Matthew. I'm in the diploma program.
Okay.
I'm Daniel. I am an exchange student.
Okay. Colombia. ¿Qué pasó, amigo? Cool, cool, cool. Okay, nice, nice, nice. I think you sent me an email, right? Yeah, yeah, yeah. Okay, well, nice to meet you. And finally,
And I'm Julien, and I'm doing systems and information biology, but I really like your offer about talking about more computational topics.
more computational stuff. Okay, so very quickly, the reason why I like computational, I am a little nerd. I used to code a lot when I was younger, so I've been coding for maybe 20, 21 years. So I learned a lot of stuff about, computational, maybe not genomics, but I've done a little bit of machine learning, mostly web, recently mostly mobile application development, but I've done all of it, okay? Why mobile? Because the money is there. And I had a company that used to develop software called RIM Labs, which recently closed. Closed because problems with partners, but that's okay. So we were profitable. We made some money. It worked a little bit. Now we're pivoting towards more mobile, private, than actually, building stuff for other people. Okay. So I do like computational stuff. Obviously, I'm not going to make it boring for the others. But if you're in genomics, you'll see that all the tools you'll use are on the computer. Okay. So we'll do that. See you next week. Don't forget the homework.